Okay, so let's see. Let's do this. Transcribe and translate a gene. I'm sorry that the computer audio is not going to come through in the recording. So I've got the closed captions on. Transcription and translation. Yes. There we go. Are the processes that all living things use to read the information in DNA and build proteins. Each gene in a cell's DNA codes for a specific protein. During transcription, cells make a copy of a gene out of mRNA. During translation, they use that mRNA copy as a blueprint to build proteins out of amino acids. These proteins perform the tasks that make living things function. That is true. The flow of genetic information from DNA to mRNA protein is central to life. Yes. Okay. Choose a gene to begin. Oh gosh. About this gene. Yeah. Here we are. Look at all that DNA. Google Translate. Somali. This is not Somali. Okay, well, whatever. Anyway, in the nucleus, transcription. Upbeat music. Inside the nucleus, RNA polymerase attaches to a gene, separates the two complementary DNA strands, exposing the gene's unique sequence of bases. There we go. Okay, so we've got our DNA here, and it's time to make some mRNA. Remember that A in DNA pairs with U in tRNA. So to begin transcription, no, I don't want to translate. Oh, good Lord. T is going to pair with A. So let's put an A nucleotide over there. G pairs with C. G pairs with C, making our mRNA. T with A. T with A. T with A. C with G. G with C. G with C. A with U. And so on and so forth. mRNA is made. Transcription is complete. When the end of the gene is reached, transcription is complete. By RNA polymerase. Wait, no, it doesn't. First, remember that RNA processing happens where we add a 5' cap and a poly A tail onto either end of the mRNA, and that prevents it from being degraded because mRNA is not a stable molecule. We need to keep it from degrading and from coming apart before the protein is made. The 5' cap also helps the mRNA find the ribosome, which is where it's going next. Also, we have splicing that happens as part of mRNA processing, and that is when the non-coding parts of the mRNA, which is called the introns, are cut and taken out, and then the remaining mRNA, the exons that do code for the protein, are spliced back together. So once again, you have a continuous strand of mRNA, and then we leave the nucleus and into the cytoplasm, where translation takes place. Yeah. Instructions in an mRNA strand are read in groups of 3 bases called codons. Each codon must be translated one after another in order to build a functioning new protein. Key structures in that process are ribosome and transfer RNA, tRNA. Each tRNA molecule has its own anticodon, anticodon, and a matching an amino acid, the building blocks of proteins. To begin translation, a tRNA molecule coded for the amino acid methionine docks with ribosome. Yes, methionine is always first. The ribosome attaches to the end of the mRNA strand. Slides along until it finds a start codon, AUG. There we go. Complementary base pairing. The start codon pairs with the anticodon on the tRNA molecule, signaling the beginning of the protein instructions. The methionine molecule is now the first amino acid in the new protein, right there. The ribosome shifts to read the next codon. Each codon is recognized by one of 20 different tRNA molecules. Each type of tRNA carries a different amino acid. The ribosome continues moving along the mRNA strand one codon at a time, matching it to a complementary tRNA. It adds each amino acid to the growing protein in just the right order. The sequence of amino acids determines the final shape of the protein and the function that it will carry out. Give translation a try. Okay. All right, so here we've got our ribosome. These guys look like mummies. Somali English. Fine, translate to English, whatever you want. Here's our codon, CAG. We need an anticodon that will pair with that, so we need GUC. That's right here. GUC, that's glutamine. Okay, cool. Out goes that tRNA. Now it's time for a new one. GCA codon. So the anticodon that we need will be CGU. CGU, there's the anticodon, and that's alanine. All right. CAC is the codon. The anticodon will be GUG. There you go. GUG. It's the anticodon. Histidine is next. Okay. So you see that the amino acids are being transferred and they're being linked together to make the new polypeptide, AAC. So the anticodon will be UUG, right here, UUG, that's asparagine. CAC, which will be GUG, again, histidine, and so on and so forth. These empty tRNAs go out and they go to a different enzyme that just puts more amino acids back on them. So they can be used again. And here we go. Yeah. When the ribosome reaches a stop codon, it detaches from the mRNA strand. Chain of amino acids folds into its final form, and the new protein is complete. This protein, aminoacyl tRNA synthetase, attaches the right amino acid to an empty proper tRNA molecule, charging the tRNA so that it can participate in protein synthesis. Oh, that's cool. Each type of amino acid has its own enzyme. Incredible processes of transcription and translation enable the cells to carry out all the functions necessary for life. There we go. All right. Stop share.